Hello and welcome back to the BHS channel, Biochemistry for Health Sciences channel. Today we'll talk about a class of molecules called proteoglycans. Proteoglycans. We mentioned proteoglycans several times in the previous videos. So today we'll talk about what is in the chemistry and biology of proteoglycans that allow them to do so many different things. So that's a question we're going to try to answer. What's so special about proteoglycans that allows them to do all kinds of things for the cells, for the body? So first of all, what is a proteoglycan? So basically these are carbohydrates. The major part is carbohydrates, and then to that carbohydrates can be attached quite a bit of protein. Okay, so if 50-60% is carbohydrate, then the remaining 40 to 50% is protein. Compare this to glycoproteins. In glycoproteins, you can see the carbohydrate is only about 10 to 15%. Okay, so some people call these proteins, I would just call these carbohydrates with attached proteins. So what basically it is, you can see shown in blue here is the protein. And attached to this protein, attached to the protein shown in blue, are the carbohydrates. And these carbohydrates are called glycosaminoglycans, or GAG, glycosaminoglycans, and those carbohydrates are shown here. And you can see the proteoglycans can exist as simple structures like this, or they can exist more attached to other carbohydrates. Like here is a long hyaluronan or hyaluronic acid, and the proteoglycans are attached to this hyaluronic acid. Okay, so you can get all kinds of complex proteoglycan structures. Now, one very important point about these carbohydrates, these GAGs, there are a variety of GAGs. You can see chondroitin sulfate, whose structure is shown here, keratan sulfate, heparin sulfate and heparin are very similar. Heparin, you're probably aware in the healthcare field, very important drug, anticoagulant. Dermatan sulfate, hyaluronan. And what we see if you look at these structures is you see this many groups, sulfate, carboxyl. You see a lot of, here's another sulfate, okay, sulfate. So many negatively charged groups. So basically GAGs are linear polysaccharide chains with repeating negatively charged disaccharides. Okay, so these disaccharides are repeated many times. Okay, and you can see disaccharides, these disaccharides are rich in negative charges. So negative, a lot of negative charges. And also note that these are carbohydrates. Okay, so you can have all kinds of shapes because they can be attached in many different ways. Beta 1,3, beta 1,4, alpha 1,4, and so on. Okay, so not only do they have negative charges, but they have a variety of shapes. Okay. And on top of that, these GAGs, they can change what GAGs are bound to a protein. They can actually change under changing physiological conditions. So that's very interesting that different types of carbohydrates could be attached to that protein to form proteoglycans that can keep on changing in their composition. So one of the reasons why proteoglycans can do so many different things for cells, for tissues, is because if you look at the biology, the location Proteoglycans are inside cells, on cells, and around cells. So for example, we have proteoglycans that are outside cells. Example, mimicin, biglycan, perlecan, decarin, certain collagens. Okay, certain collagens are proteoglycans that are outside cells. And you can see what is the major carbohydrate they contain: keratin sulfate, chondroitin sulfate, dermatan sulfate, and so on. And some of these proteoglycans, they exist as much larger structures, aggregates, because they can bind hyaluronan, or they can bind lectins, which are proteins. So here is a hyaluronan which is attached to many proteoglycans. So these are also extracellular, so we'll just call them extracellular aggregates. So they're basically aggregates of these ones coming together and joining together to form very, very large structures. They're extracellular too, mostly you can see in the connective tissue. So these proteoglycans are not only extracellular, but they could also be attached to the plasma membrane. They are transmembrane. They are attached to the plasma membrane. Okay, so you can see a lot of these can be attached to plasma membrane. Examples: syndecan, glypican, beta-glycan, phosphorcan, and again you can see the glycosaminoglycans such as chondroitin sulfate or heparin sulfate that are attached to these proteoglycans. Very interesting, recently they've also discovered that there is actually a proteoglycan inside cells, intracellular, and that's called sirglycin. It's an intracellular proteoglycan, very rare. This is one example that has been seen inside cells, and we'll talk about it in a few seconds. So as far as the biology part, proteoglycans are everywhere around cells, so it's not surprising they can do many, many, many things for cells. What about the chemistry part of proteoglycans? What's in their chemistry that allows them to do so many different things? So here's some of the major features, chemical features that help them carry out their functions. First of all, these carbohydrates, have a lot of hydroxyl groups. We talked about carbohydrates before. And hydroxyl groups, water-like hydroxyl groups. So not only are there a lot of hydroxyl groups on the carbohydrates have negative charges. Water loves positive or negative charges. So because of these features, these carbohydrates attract a lot of water. And so the tissue becomes filled with water, and that gives the tissue mechanical strength. It keeps the tissue hydrated because this water are quite strongly attached to these carbohydrates. And water is, when you have water in a tissue, it also acts as a shock absorber. So this attraction of water allows proteoglycans to do quite a lot of good roles for tissues. And the other thing about these proteoglycans is that, again, partly because of the fact that they have so many hydroxyl groups, and hydroxyl groups can form hydrogen bonding. So you can see a lot of hydrogen bonding between these molecules. Okay, and that can lead to the formation of gels. So more sort of in between a solid and a liquid. So because of all this cross-linking by these weak forces like hydrogen bonding, these proteoglycans can give the tissue a gel-like consistency, and that allows the tissue, or gives the tissue, mechanical strength, and also acts as a protective barrier, because it's not easy for a microbe to cross through this gel to go over the other side. The third chemical feature of these proteoglycans is what we talked about earlier. The carboxyls and the sulfates and all give these carbohydrates, these GAGs, have a very high anionic charge density. That means they, in a small part of they have a lot of negative charges. Okay, high ionic charge density. And these highly negative charged areas, one of their major function is they bind and store cationic signal molecules. So chemicals that are signals, and we'll talk about this under cell signaling, and these chemicals tend to have a net positive charge. Okay, so these chemicals actually get attracted. So you can see here a cationic signal molecule getting attracted to these negative charges. So they get attracted by this electrostatic or ionic interaction. And these signals play a very important role in cell division, which is very important for tissue repair as well as for cancer. So a lot of cell signaling that happens because of these proteoglycans. So not only is their shapes, the shapes themselves have so much information that allow them to be used as cell signals, but then they also trap other cell signals that are cationic, positively charged. So basically around the cell, you have a lot of signals ready to tell the cell what to do. So let's quickly go through a few examples. The intracellular proteoglycan glycine, very rich in mast cells as well as other cells. One of the functions of mast cells is that it stores heparin. Has a lot of heparin. Heparin is the GAG, the glycosaminoglycan, that is found in these proteoglycans that are the glycine, very rich in heparin. And the mast cells have a lot of signal proteins, such as cytokines, chemokines, growth factors. And once again, these signal proteins are basic proteins. That means they have a net positive charge. And therefore, these net positive charge proteins get attracted to the negatively charged heparin that is part of the proteoglycan glycine stored inside the mast cell. Just to note that the small molecule histamine is also an amine, a cationic, a positively charged chemical that is very rich in mast cells. The transmembrane proteoglycans, just a few examples and what they do. Syndecan, that has these GAGs, chondroitin sulfate and heparin sulfate. Syndecan connects cells to the extracellular matrix, so the ECM connection connects the cells to ECM. They bind all kinds of growth factors that are cationic, positively charged. These proteoglycans act as endocytosis receptors. For example, they can act as receptors for various lipoproteins. Of course, cancer and extracellular matrix and proteoglycans is a big connection. So they can promote cancer growth, they can promote the spread of cancer. And then there is this shedding of syndecan. So basically what that means is this part of the syndecan can be removed, can be broken away and become free away from the plasma membrane. And interestingly, this syndecan can enter the cell and actually go to your DNA, to your nucleus, and affect the cell signaling right there itself. Okay, so the syndecan can affect very important enzymes involved in DNA metabolism, such as DNA topoisomerase and HAT histone acetyl transferase. all of which we'll talk about in future videos. Another transmembrane proteoglycan is beta-glycan, which is rich in chondroitin sulfate and heparan sulfate. This is actually a receptor for a signal called TGF, transforming growth factor. It's a growth factor. It's actually a receptor. And so this signaling is very important for development, fetal growth. And of course, whatever is important for fetal growth, unfortunately, is also used quite often by cancer. Because what are cancer cells? They're basically very fast dividing cells, and that's what we see in a fetus. So a lot of tricks, a lot of things we see in fetal growth is also something we see quite often in cancer. Phosphocan is another transmembrane proteoglycan, rich in chondroitin sulfate, and that again helps cells bind other cells, cell-cell adhesion, and plays a very important role in the development of the human brain, phosphocan. Glypican, another transmembrane proteoglycan, plays an important role in the growth of blood vessels. Okay, formation of blood vessels, new blood vessels. And as you know, blood vessels are so critical because it supplies energy. So angiogenesis, glypicans play, not surprising, a very important role in angiogenesis. And angiogenesis, of course, is very important for fetal development as well as for growth of cancer cells, tumor growth. Then we have the extracellular, the proteoglycans that are found outside the cells. And here is some examples of this. Perlecan is very close to the cell, found very, very close to the cell, and also sometimes in the basement membrane of many cells. It has again many different functions, and we can understand why because of the biology and the chemistry. Cell adhesion, skin and bone formation, mechanical properties of cartilage and blood vessels. It helps modulate growth factor activity. It's involved in lipoprotein binding and metabolism, involved in tumor angiogenesis, growth of new blood vessels, regulates nitric oxide synthase, very important for blood vessels, involved in neurons, function of neurons, inflammation, tissue repair, autophagy. So many functions of these proteoglycans. They play a role in so many different activities. Not surprising because they are found everywhere around all the cells and connective tissue. So it's not surprising that they play many different roles in many different tissues. Some of the collagens are also extracellular proteoglycans. Okay, so these are predominant in the basement membrane of epithelial cells and blood vessels, involved in lipoprotein metabolism, blood vessel growth, adipocyte and skeletal muscle structure, and so on. And then the class of extracellular proteoglycans called hyaluronan-lectin binding proteoglycans. So the hybrid word is hyaluronan-lectins, so hyalectins. These are sometimes called hyalectins. So these are the ones that include agrican, versican, brevican, neurocan. These are the ones that can bind, their binding sites for hyaluronan. So this, for example, can bind here, this can bind here, this can bind here. And when they do that, they form very, very large structures that we call aggregates. Okay, so agrican is the major the major proteoglycan of cartilage that gives cartilage its mechanical strength. Versicolin is involved in cell adhesion and migration during development, as well as in inflammation. It's involved in leukocyte migration. Brevicin and neurocin are very important in the brain. They're important for brain development as well as for brain repair after injury. So just to summarize, proteoglycans are very important molecules in the human body. Large branching structures allows multiple weak forces for interactions with all other kinds of molecules. High anionic charge, negative charge allows strong non-covalent interactions with cationic molecules such as growth factors, chemokines, cytokines, and so on. Allows the cells to bind to extracellular matrix and to one another. Modulate mechanical properties due to gel-like property of these proteoglycans. They attract a lot of water, therefore keeping the tissue well hydrated, giving the tissue its turgor pressure, shock absorbing property, maintain the size and shape of the organ. Again, because of the negative charges, they can sequester a lot of cationic proteins. And we'll talk about PI, isoelectric point, very important part of proteins in a future video. So proteins with a PI greater than 7.4, many of these are very important signaling proteins such as growth factors. So these proteoglycans help tissues trap and store these growth factors, chemokines, cytokines, and so on. And in lipoprotein metabolism, we'll come across this enzyme called lipoprotein lipase, LPL, which is a high PI, PI8. And that lipase basically sticks to the endothelial surface and it plays a very important role in the metabolism of lipoproteins. So it's not surprising that when the clinician administers intravenous heparin, which is negatively charged, that negative charge heparin can actually release this lipoprotein lipase into the bloodstream. Okay, so it's kind of interesting. So here you have an endothelial cell, your LPL is stuck to the endothelial cell. So the proteoglycans here is negatively charged, the LPL is positively charged, but when you give heparin, which is high negative charge, that heparin kicks out the LPL, and the LPL, the lipoprotein lipase, gets out into the bloodstream. So just to connect the previous video that we call molecular recognition, so you can see this is a great example. You see the negative charges on your proteoglycans. Let's say these were endothelial cells, right? Endothelial cells. And these negative charges will attract proteins that have a net positive charge, such as your lipoprotein lipase. You see, so that's molecular recognition. So the force that's holding this together is electrostatic. Electrostatic. Okay, so information in the form of these forces that we talked about earlier, electrostatic, hydrogen bonds, hydrophobic forces. And then when you inject heparin, which is negatively charged, okay, the heparin will basically stop this attraction between these two, and the lipoprotein will now basically get attracted to heparin and come out into the bloodstream. Okay, so this is a well-known phenomenon that heparin administration releases lipoprotein lipase into the bloodstream. So once again, a good example of molecular recognition. So hopefully little by little we are trying to get our concepts together so that we can start seeing the bigger and bigger picture, which is very critical for healthcare professionals. That's all for now. Until next time, let's say goodbye and have a very, very good day.